All right, developmental biologists talk a lot about genes, and developmental biologists do a lot of genetic analyses. And so, as a kind of a background for some of that stuff, we're going to talk about insulin. And we're going to talk about insulin because it comes up as an example in the course, and also because students have a good kind of conceptual understanding of insulin. Insulin is a protein hormone that is made in every vertebrate. It gets secreted into the blood, and it has a job in metabolic homeostasis. Insulin is made in the pancreas in every vertebrate, and so we're going to zoom in to the pancreas, and we see that insulin is made in only a subset of the cells, the beta cells. What does that mean? That means that although every cell in the body has the insulin gene is only turned on or active in the beta cells. What does that mean? That means that RNA polymerase binds to the insulin gene only in the beta cells, and RNA polymerase transcribes the insulin gene into primary RNA transcript, which is spliced and processed into mature RNA in the nucleus of the beta cell, is exported into the cytoplasm, and then translated by ribosomes on the rough ER into insulin protein, which is processed and then secreted into the blood and diffuses throughout the body. We can zoom in on the insulin protein molecule. Like any protein, it has a three-dimensional tertiary structure, and that structure confers its function. The function of insulin is to diffuse through the blood and bind to the insulin receptor on the surface of target cells. It's through that three-dimensional chemical structure that insulin protein function occurs. We can look at the structure of any protein molecule and see that it's composed of chains of amino acids, and we can grab those chains and straighten them out and look at what's called the primary amino acid sequence, which is a linear sequence of amino acid chains, which are joined together by covalent bonds called peptide bonds. And that's true for any protein, and each protein has a unique amino acid sequence, a unique primary amino acid sequence. There are 20 common amino acids, and so that's what these colored balls are each of the 20 amino acids chained together in this distinctive primary amino acid sequence. We can represent that primary amino acid sequence as a series of letters by using a single letter abbreviation for each amino acid, and that looks like that. That's a powerful way to store genetic information, because we can store it just like words in a book or in a library, and we can search through those books the same way we do word search on a computer. And we're going to look at how that's done. First, we look at the fact that the insulin protein sequence information is collinear with DNA sequence information in the insulin gene. The insulin protein is composed of a chain of amino acids. The insulin gene is composed of a chain of nucleotide letters, A, G, C, T. This is a good example to look at to see that, in fact, the human genome, enough information to code for one human, so the amount of genetic information you got from your mom or your dad, one human genome is actually really long. It's about 3 billion letters or nucleotide letters long. So that that familiar DNA double-stranded molecule has to be folded into tight structures that are composed of DNA plus protein and called chromatin, and that chromatin is packed away into each of the chromosomes in the nuclei of each of our cells. We can make comparisons between that stored information that's available on computers. And so if we could, for example, take Nancy's insulin gene and search through Bill's genome to find a match, just the same way we do a word search on a computer. That word search program is called BLAST, and so to make this comparison, we do something called a BLAST search. This is a website that just shows how huge amounts of DNA information are stored and can be searched with a variety of tools. The one we'll focus on in this course is this BLAST search, which is like a word search. We line those up, and what was done is that although the single letter abbreviation of amino acids is shown in this figure, what's usually searched is actually the DNA. And so Nancy's insulin gene was used as what's called the query sequence to search through an entire human genome, like Bill's for example, and find the best match. Here's what an output looks like from a blast search. That upper colored bar shows that there's an excellent match between Nancy's insulin gene and Bill's insulin gene. The red bar shows that it was a good match throughout. Down below, we see that what was identified as the best match is Bill's insulin proinsulin precursor, the insulin gene, and that this e-value shows what the likelihood was that that match would occur by chance. And it's a really low likelihood. It would occur by chance 1 out of every 8 times 10 to the minus 62 times. So there's a really good match. What this figure doesn't show is that the BLAST program keeps track of all the matches, rates them all, and ranks them all, and we're just looking at the best match. There's this direct match between the insulin genes. We can do that same BLAST search and look at any 2 people, and we can find that the insulin genes between us are slightly different. Different versions of the same gene are called alleles, and we'll talk a lot about alleles in this course, but we'll talk about them in a slightly different way than this introduction, and we'll talk about that. We can also do BLAST searches and use the human insulin gene to search for matches in other organisms. And when we do that, we find this, that in all vertebrates, we find the best match, and it's a very, very strong match between human insulin, is an insulin gene in each of those organisms. This shows the sequence match. So that's what we get out of BLAST search, is just the sequence match between these different species. This is how developmental biologists think about that kind of comparison. They start out seeing this similar amino acid sequence, and then they do further studies to find out what the function of the protein is in each of the species. And we find that the similar sequence gives a similar function. So the sequence confers the structure of the protein, and each of these proteins folds up to be something like a screwdriver. The insulin gene in chickens codes for a somewhat different protein than in humans, but their structures are similar, and basically we're talking about 2 slightly different screwdrivers, literally. The insulin gene codes for a physical protein that is just like a tool, and its structure has a job to do. The genes the proteins might be slightly different, and so maybe we can't just use the insulin protein from chickens and humans because there might be enough difference that our screwdrivers don't match up. But by studying the function of the protein in each of the species, we see that they're doing the same job. That leads to this interpretation, that we have the best match, the human insulin gene in chickens is what we call the chicken insulin gene. And the evidence is from both the structure, the match, and also the function. Studies that show that the insulin gene codes for protein that's doing the same functional job. The interpretation is that those matches occur because we evolved from a common ancestor that had an ancestral insulin gene, another screwdriver that looked quite a bit like the modern insulin proteins, but had some differences. And we'll talk a lot about that concept in this course, and this concept is given a term. And so orthologs are 2 genes in different species that share a common ancestral gene. Again, that's an interpretation of the history of the relationship, but we'll use that concept a lot in this course. And again, the orthologs are genes in different species. So the human insulin gene has an insulin gene ortholog in a chicken. We can build evolutionary trees using that kind of logic. So this just shows that historically, Charles Darwin created what's called phynetic trees. Phynetic trees are based on observed similarities, like these all have hair. Phylogenetic trees that we'll talk about in this course are based on evolutionary relationships. relationships like the story of the ortholog. And the idea is that phylogenetic trees are based on this. These all have closely related insulin genes. Again, closely related based on DNA sequence, and then also based on insulin protein function in those two species. There's this simple answer that we've just gone over here. And the evidence for phylogenetic trees based on that simple description is huge because it's true for every gene. And we'll talk about a lot of genes in this course and a lot of relationships between species in this course. We won't have time to talk about the detailed answers of the arguments that these are insulin gene orthologs. But in every case, it's important to remember that there's a blast search that showed a similar sequence between genes, and then they're followed up by functional analyses showing that the gene products do comparable jobs in the two different species. So we have this orthologs are two different genes that share a common ancestor, and that's the take-home message from this. We'll use that term a lot in this course and that concept a lot.